Hi guys, I'm Mithila Rajapakse. Welcome to our YouTube channel Gamsapi. And under this set of videos, we'll be studying about the reproduction process that occurs between living organisms. And further, we'll be studying about the human reproductive system, both male and female. And this reproduction section comes under unit number 5, animal form and function. So let's see what comes under this video. First, we have the modes of reproduction seen among animals. First is the asexual reproduction, and under that we'll be studying about budding, fragmentation and regeneration, and parthenogenesis. And next we have sexual reproduction, and under that we'll be studying the gamete formation process, unisexuality and bisexuality, and the fertilization process. And next we'll be studying about the significance of sexual and asexual reproduction. And as the second part of this video, we will be studying about the human male reproductive system. And under that, the structure and function of the human male reproductive system, and the spermatogenesis process, and the accessory glands associated with the male reproductive system, and the hormonal control or the hormonal regulation of the male reproductive system. So let's start the lesson. First of all, the modes of reproduction seen among animals. Reproduction is a biological process through which a new generation of individuals is produced from the existing organisms. So this is the basic definition for reproduction. A new generation of individuals is produced from the existing organisms. Two modes of reproduction are found among the animals. The first is asexual reproduction and sexual reproduction. So we'll be studying about these two, asexual and sexual reproduction, in the next few slides. For the most animals, reproduction is mainly or entirely sexual. So sexual reproduction is the most commonly found mode of reproduction among living organisms. Several forms of asexual reproduction are also seen, especially among invertebrates. So invertebrates mostly have asexual reproduction, and in addition, vertebrates also sometimes have asexual modes of reproduction. Then next, talking about asexual reproduction. Asexual reproduction is a mechanism through which new individuals are generated from a single parent without the fusion of egg and sperm. So this is the unique characteristic of asexual reproduction. There is no fertilization, or there is no genetic combination occurring between the egg cell and the sperm cell. Asexual reproduction relies entirely on mitotic cell division. Here, mitosis is the process where asexual reproduction occurs. Asexual reproduction allows the rapid multiplication of individuals from single parents. So this is an advantage, the rapid multiplication of individuals. It takes less time when compared to sexual reproduction. Hence, no time or energy spent in searching the mates for the reproduction, because only one organism the one parent organism is required. The produced offspring are genetically identical to each other and to the single parent. So, as I said before, mitotic cell division is the basis of asexual reproduction. So, you might be aware that when mitotic cell division occurs, the newly produced daughter cells are genetically identical to each other. They have the same genetic composition, and also to the single parent. They have the same genetic composition as the initial single parent. Several methods of asexual reproduction are found among invertebrates. So they are budding, and then next we have fragmentation and regeneration, and then parthenogenesis. So starting off with budding. What's budding? Budding is a form of asexual reproduction in which individuals arise from outgrowths of the animal. Example, hydra. A localized mass of mitotically dividing cells develops into a small hydra which can eventually detach from the parent. So here a small outgrowth first forms from the parent animal. Here they say a new individual arises from the outgrowths of the animal. So an outgrowth first rises, and this outgrowth develops, and after a certain stage it detaches from the parent organism. So at that moment the new organism can survive on itself without depending on the parent. So hydra is an example for the organisms which carry out budding. Then next, fragmentation and regeneration. This is a form of asexual reproduction which involves breaking of the body or part of the body into several pieces, followed by the growth of a separate individual from each piece. And each fragment develops into a complete animal by regrowth of lost body parts, and this is the regeneration process. Now here, first you have the parent organism, and this parent organism breaks down itself into let's say around 3 or 4 pieces, or not the complete body of the parent, or maybe a part of the body. So after breaking down into several pieces, each of these pieces develops into a new individual. So they grow the missing parts of the that considered section, and each part develops into a new individual. So the examples are annelid worms, numerous sponges, and cnidarians. So cnidarians are the coelenterates. Here you can see the budding process of an hydra. First, a tiny bump appears on the parent hydra. So this is the tiny bump which occurs in the parent hydra, and the parent hydra develops the bud, and then the bud gradually grows. So here you can see the bud develops into a new hydra, and the bud breaks off to become an independent hydra. So at this stage, this new individual hydra can survive on itself without this support of the parent. So this is the budding process of hydra. And next we have the fragmentation and regeneration of planaria. So here you can see the parent planaria, and it divides itself into 3 sections. So this middle part goes here, and these are the missing parts of the planaria. This is the head and the tail, and it grows the head and the tail to form this new planaria. And similarly, the head develops into another planaria by completing this part, and the tail develops into another planaria by completing the anterior section. So here you can see this, from this single parent, 3 new identical planaria are formed. So this is the fragmentation and regeneration in planaria. So this growth of this missing part is the regeneration process. Fragmentation is this breaking part. Next we have parthenogenesis. This is the next form of the asexual reproduction. This is an unusual form of asexual reproduction in which an egg develops into a complete individual without being fertilized. So here this individual can be either haploid or even at certain times diploid organisms are also produced by parthenogenesis. Among invertebrates, parthenogenesis occurs in some animals such as bees, ants, amphibians, and wasps. The progeny can be haploid or diploid. From this parthenogenesis process, the organism can be haploid or diploid. Now, if an egg develops into an individual, it will be haploid. If the parent directly carries out mitotic division, the progeny will be diploid. In honeybees, males, they are the drones, are fertile haploid adults that develop through parthenogenesis. So remember, although they are haploid, they are fertile. Whereas females, including both sterile workers and the fertile queen, are diploid adults. They develop from the fertilized eggs. Parthenogenesis has also been observed very rarely among some vertebrates. So lizards and fish are vertebrates. They also carry out parthenogenesis process. So here you can see this parthenogenesis of honeybees. First you have the haploid male bee. It carries out mitosis to produce the N type of sperm cell. And then you have the diploid type of the queen bee. It carries out meiosis to produce an egg cell. This is also an N type of haploid cell. So here you have the N type haploid egg cell and the N type haploid sperm cell. So it undergoes fertilization to produce a diploid cell, and from this diploid female, 2 N workers and the sterile female will be formed, and the queen, which is a diploid organism, will also be formed from this fertilized female. And on this path you can see the meiosis of the queen bee to produce an N type of egg cell. Now here this is the parthenogenesis process. Here no fertilization occurs, and which results in the production of an haploid type of organism. This is the male, the haploid drone. So this is the parthenogenesis which occurs in the honeybees. Now moving on to our next form of reproduction, the sexual reproduction. Sexual reproduction is a mechanism through which a new individual is developed from a diploid zygote. As a result of the fusion of 2 haploid gametes, which are the sperm and the egg, which are produced by two individuals, the male and the females respectively. So this zygote is what is produced after the fertilization of the male and female gametes. The gametes are haploid, these are the sperm and the egg, and they are produced by two individuals, the male and the female parents. So because two haploid cells join, the diploid cell, the zygote will be a diploid 2N type of cell. The female gamete, which is the egg, is large and non-motile, whereas the male gamete, the sperm, is generally much smaller and motile. So we'll be learning this in the slides to come. The egg is a non-motile structure. It's a large and non-motile structure, and the sperm is much smaller. And here the sperm swim towards the female gamete, which is the egg. The fusion of the female and male gametes forms a diploid cell called the zygote. I just explained that. So here the female and male gametes, which are haploid cells, combine to form the diploid zygote. The animal that develops mitotically from a zygote can in turn give rise to gametes by meiosis. So it can then, in the next stage, this newly formed zygote after development can carry out meiosis, so that gametes will be produced by it. So sexual reproduction will be promoted in those organisms. For the most animals, reproduction is mainly or entirely sexual. So we talked about that before. For most animals, reproduction is mainly or entirely sexual. So talking about the gamete formation process, you call this the gametogenesis process. In animals, reproductive cells called gametes, these are the sperms and the eggs, are the vehicles that transmit genes from one generation to the next. So you may be knowing the sperm and the eggs contain the genetic material of the individual. So that's why they say the vehicles that transmit genes from one generation to the next. Each gamete is haploid, unicellular reproductive cell. It has only a n number of genetic material, so that when the combination of the female egg and the male sperm occurs, the final resulting individual will be a diploid organism, a 2N type of organism. Gametes in animals are formed by meiosis in special organs called gonads. So there are special organs known as gonads, and this meiosis takes place in these gonads, so that it will result in the production of the sperms and the eggs. Bisexual organisms and unisexual organisms. In a bisexual organism, or you call them the hermaphrodites. Hermaphrodites are the bisexual organisms. One individual has both male and female reproductive structures, and therefore is able to produce both male and female gametes by the same individual. Now here, a single individual consists of both the male and female reproductive gonads. So a single individual can produce both male and female gametes due to the presence of these both reproductive structures. For example, earthworms. Earthworms are hermaphrodites or bisexual organisms. In unisexual organisms, or you call them dioecious organisms. So these bisexual organisms are known as monoecious organisms. One individual bears either male or female reproductive structures. So here, when you take an individual, the individual either consists only the male or else only the female. They never have both the male and female structures in a single individual. Therefore, production of male or female gametes occurs in separate organisms. For example, in humans. We have two distinct sexes as the male and female, and so here they carry out the gametogenesis process individually in a male or in a female separately. So here you call this dioecious because di means two. So here you have two individuals for the two different opposite sexes. Then the fertilization. Fertilization is the union of the egg and the sperm. So they are the gametes culminating in the fusion of the nuclei is called fertilization. So fertilization is the, in brief, if you say fertilization is the process of fusion of the nuclei of the egg and the sperm. So here N-type nuclei and the sperm contains an N-type nuclei. So the diploid zygote will be having a 2N type of nucleus. Fertilization can be either external or internal. So now let's see what's this internal and external fertilization. External fertilization occurs in aquatic environments In species with external fertilization, the female releases eggs while the male releases sperm into an external environment, and fertilization occurs in water. So here the fertilization process occurs outside the bodies of both the male and the female, in the immediate environment that they are surviving in. A moist habitat is almost always required for external fertilization, both to prevent the gametes from drying out and to allow the sperm to swim towards the eggs. So here this external environment must be a moist environment, or in other words, it must have water. So the reason they specify for the environment to be moist is to prevent the gametes from drying, or to prevent desiccation of the gametes. So if the gametes dry out, the gametes will be died. The moist habitat prevents this from happening, and also to allow the sperms to swim towards the egg. So when the gametes are released to a fluid medium, the sperm can swim towards the egg, and also the egg can move a little bit towards the direction which the sperms are produced. So this is a trophic movement which occurs in response to chemical signals. Many invertebrates, amphibians and most bony fish, so these organisms carry out external fertilization. In internal fertilization, sperms are deposited in or near the female reproductive tract, and fertilization occurs within the female reproductive tract. Example, insects, reptiles and mammals. So here this internal fertilization can mostly be seen in terrestrial environments. The reason is, as I said before, external fertilization requires a moist environment. So in terrestrial organisms, finding an aquatic environment to carry out external fertilization is quite a bit difficult task. So as an adaptation, this internal fertilization has been included into the species. Internal fertilization is an adaptation that enables sperms to reach an egg even when the environment is dry. That's what I just said. The male copulatory organ delivers sperm, and the female reproductive tract often has receptacles for storage and delivery of sperm to mature eggs. Sorry. So we'll be talking about this process, how the fertilization process occurs, in the future videos to come. Until that, this would be sufficient for you. Internal fertilization is typically associated with the production of fewer gametes than external fertilization, but results in the survival of a higher fraction of zygotes. Now, when external fertilization occurs, the gametes are released to the environment. So there is a bigger chance of these gametes in dying, or let's say the fertilization process will not be very productive when it's released into the environment, because it's not a favorable or enclosed environment. But in an internal fertilization process, the environment is enclosed, so there is a much probability or higher chance of a sperm meeting the egg and carrying out fertilization. Internal fertilization often provides greater protection to the embryo, so the embryo develops within the female body. Many animals show parental care. Internally fertilized eggs of birds and other reptiles possess shells and internal membranes that protect against water loss and physical damage. So when you have shells, so hard shells, rigid shells and internal membranes, water loss is prevented, no desiccation occurs, and also physical damage is prevented. These shells and the internal membranes acts as a physical barrier. Some retain the embryo for a portion of any development within the female reproductive tract. So for example, in humans, generally 9 to 10 months the embryo is surviving within the body of the mother until the certain stages of development are passed by the embryo. So then moving on to the significances of asexual and sexual reproduction. Asexual reproduction and sexual reproduction are important biological processes to ensure existence of species. Asexual reproduction allows rapid multiplication of individuals from a single parent. Hence, no time or energy is needed to be spent in searching mates for the reproduction. So here this is an advantage. rapid multiplication and the requirement of a single parent. So the time and energy spent and searching for mates for asexual reproduction is less compared to a sexual reproduction. In asexual reproduction, there is no or very little genetic variation within a population, as the produced offspring are genetically identical to each other and to the single parent. Therefore, asexual reproduction would be more advantageous in stable, favorable environments, as it can propagate successful genotypes specifically. So what does this part explain? First, they say there's little or no genetic variation. So as I said before, mitosis occurs here. So this very little genetic variation can be due to certain mutations which occur during the cell division. However, mostly there is no genetic variation occurring. Asexual reproduction would be more advantageous and stable or favorable in favorable environments, as it can propagate successful genotypes specifically. So once an organism gets adapted to a certain environment, through mitosis, the same set of genes will be transferred to the new generation. So that new organism will also be able to successfully survive in that environment. But if in sexual reproduction, there is a change in the genetic composition. So sometimes the newly formed organism might not be able to survive in the environment that the parents survive, because there is a change in the genetic composition. But in asexual reproduction, due to mitosis, such an occurring would not take place, because the genetic composition of the parent and the new offspring is identical. However, any mutation in the parent cell can cause harmful effects on the survival ability of the offspring in changing environments, as well as I just explained. If there is a harmful mutation in the organism, environment changes could be deadly to the individuals in the population. So if a change in the genetic composition occurs due to mutation, the environment might be deadly. So the organism might not be able to survive in the environment that its parent was able to survive because of this mutation or the change in the genetic composition of the new offspring. Unlike asexual reproduction, sexual reproduction results in the production of a unique offspring by combining genetic materials from two parents. So that's the male and the female. Meiotic recombination during sexual reproduction helps produce varied genotypes. So here you might have already studied this. Meiotic recombination during sexual reproduction. During the meiosis process, due to the independent assortment of chromosomes. Independent assortment of chromosomes allows the genetic characters to be mixed. In addition, during the fertilization process, as you know, many sperms are produced. So only a single sperm will penetrate and fertilize the egg cell. We'll be studying about this in the later videos. So here, because of these, there will be a varied set of genotypes produced from one generation to the next. In addition, during this meiosis process also, a process known as crossing over occurs. Here, the genetic material are exchanged during the crossing over process. So this will also contribute to produce a varied genotype in the new offspring. Unique gene compositions formed during sexual reproduction would be advantageous, as this can enhance reproductive success and survival of a species in changing environments. So this is the adaptation and evolution process of the organisms. With the as the generations continue, new characters will be introduced due to mutations in the genetic material, and this can enhance the reproductive success and the survival of a species in the changing environments. Beneficial gene compositions arising through recombinations may speed up adaptation. During sexual reproduction, shuffling of genes might allow a population to clear sets of harmful genes more readily, which would enhance the survival of the species. Now, this also occurs during sexual reproduction. Here, if there is a harmful gene during sexual reproduction, during the meiosis process, this harmful gene can be removed, because as I said before, characters are being exchanged. So during the fertilization process, maybe the sperm containing this harmful set of gene will not fertilize the egg. So from there onwards, the new offspring will not have this harmful characteristic which was present in the parent. So that's the end of our first session. So this next latter part of our video, this is on the human male reproductive system, and under this we'll be studying about the male reproductive system in detail, its structures and functions, and the hormonal regulation of this male reproductive system. So talking about the structure and function of the human male reproductive system. Main internal reproductive structures of a male consist of gonads, or the testes, which produce sperm and the reproductive hormones. Epididymis, which store mature sperms, axillary glands that secrete products required for sperm movement, and ducts that transport the mature sperm and glandular secretions. So these are the components of the human male reproductive system, and we'll be studying about each of these structures in detail in the next few slides. The external reproductive organs of a male are the scrotum and the penis. So all other structures are found within the body of the human male. These are the two structures which are visible to the outside, the scrotum sac and the penis. So here you can see the gross structure of the male reproductive system, and for orientation some non-reproductive parts are also being labeled. So here you have the seminal vesicle. This is a vesicle present in the male reproductive system, and then the prostate gland. This is the prostate gland, and the ejaculatory duct. This is the short ejaculatory duct, and the bulbourethral or the Cooper's glands. This one, and this is not important. The spermatic cord. These are the spermatic cord, or you call this a spermatic duct, and then you have the epididymis. And then the scrotum sac. This is the scrotum sac. This is present externally to house the testis and the epididymis like structures. And this is the testis. And then you have this urethra. This is the urethra, the path which urine and semen passes through. And this is the glans penis, the tip of the penis. And then the prepuce or the foreskin. And this corpus cavernosum. This is a tissue, the erectile tissue present in the penis, and this structure is the penis. And then you have the corpus sponginosum. This is also an erectile tissue. Then you have this pubic symphysis. This is a bone. This is not a structure of the reproductive system. And then you have the urinary bladder. This stores urine. This is also not a structure of the reproductive system, but urine passes through the urethra, and so does the semen as well. And then here you have the ureter. That's a part of the urinary system. And then you have this vas deferens or ductus deferens, which initiates from this spermatic cord. Here you can see this epididymis, and this moves up like this, and then from here it turns, and then you have this vas deferens, and here it joins and releases its the parts move to the urethra. So we'll be learning about this all these structures in detail. So that's a general idea of the structures of the male reproductive system. Scrotum is a pouch formed by a fold of the body wall. Scrotum is divided into two compartments. Each compartment contains one testis, one epididymis, and part of the spermatic cord which suspends the testis in the scrotum. So here you can see this scrotum sac, and inside this you have a single testis. It is partitioned into two, and a single partition consists of a testis and one epididymis and part of the spermatic cord, this part. So the other compartment also consists all these structures, the testis and the epididymis and the section of this spermatic cord. The testis. The testes are found within the scrotum, which allows the maintenance of the testis temperature 2 degrees Celsius below the core body temperature. Now the reason is sperms are produced properly only when the testes are cooler than the rest of the body. So that's the reason why this scrotum, the scrotum with the testis, is situated outside the body. So when it's situated outside, the temperature will be about 2 degrees Celsius less than the core body temperature. This facilitates the spermatogenesis process. The testes develop in the abdominal cavity and descend into the scrotum just before birth. So we'll be studying about that in the later videos. At the end of the third trimester, this testis descend into the scrotum, which was present within the abdominal cavity. The testes are cooled by their position outside the abdominal cavity and the thin covering of the scrotum. So the scrotum has a thin covering. So the heat present within it will be dissipated outside and a cool environment will be maintained. Each testis contains many lobules. Within each lobule, there are highly coiled or convoluted loops known as seminiferous tubules. Sperms are produced within these tubules. Different cells that undergo spermatogenesis are surrounded and connected to special type of supporting cells known as Sertoli cells. Now these Sertoli cells are present extending towards the lumen or the center of these seminiferous tubules. These provide support and assistance to the developing sperms. Sertoli cells extend from the wall of the seminiferous tubules towards the lumen. These cells secrete the hormone inhibin. Now these cells are the Sertoli cells. Sertoli cells release or secrete inhibin and nourish and provide attachment for cells that are in different stages of spermatogenesis. So different stages are present in the spermatogenesis process. So the Sertoli cells provide assistance and attachment during this development of the cells into sperms. Group of cells known as Leydig cells or the interstitial cells are present in the connective tissue between the seminiferous tubules. So they are found between in the connective tissue between the spaces of the seminiferous tubules. Leydig cells secrete the hormone testosterone and other androgens after puberty, which promotes spermatogenesis in the tubules. So Leydig cells secrete testosterone and androgens, and they promote spermatogenesis in the tubules. Tubules in the sense seminiferous tubules. The seminiferous tubules combine to form a single tube or the duct at the upper part of the testis. That's what we call the spermatic duct. So here you can see the cross section of a testis. First of all, you see this cross section of a seminiferous tubule, and here you can see these various stages of the development of the sperm. Here you have the germinal epithelium and the progressive stages until the sperm is formed at the center. And these are the supportive Sertoli cells extending from the wall of the seminiferous tubule towards the lumen. They provide support and attachment and nourishment for these sperms to various levels of various stages of development of the sperm. And here you have these Leydig cells. These are the Leydig cells found within this connective tissue, which is present between these seminiferous tubules. So this is the cross section of the human testis. So about these various stages: spermatid, the secondary spermatocyte, the primary spermatocyte, and the spermatogonium. We'll be learning about this in the next few slides and the relevant process. Epididymis. Epididymis is the tightly packed mass that is formed by the repeatedly folded long duct originated from the combined seminiferous tubules in the testis. From the seminiferous tubules, the sperm pass into the epididymis. So once these produced sperms are released from the seminiferous tubules, they come outside of the testis and collect in this epididymis. This epididymis is a repeatedly folded long duct. So here the sperms, since it's very long, about 6 meters in length, so this epididymis is 6 meters in length, it takes about 3 weeks for a sperm to travel the length of this duct. During this time, the sperm becomes mature and motile. So here the sperm obtains maturity and becomes motile because the sperm has to swim towards the egg and also through the path of the male reproductive system. Matured sperms are stored within the epididymis until ejaculation. So this occurs as a place for storage as well before ejaculation. Now ejaculation is the process of release of the sperms from the male reproductive system towards the female reproductive system. the vas deferens, ejaculatory duct, urethra and the penis. During ejaculation, sperms are propelled from each epididymis through a muscular duct. This is the vas deferens. Each vas deferens, and from each epididymis, so each epididymis consists of each vas deferens, and from both the epididymis, sperms are released to their each individual vas deferens, and they extend around and behind the urinary bladder, where it joins a duct from a seminal vesicle, forming the short ejaculatory duct. So this vas deferens and the short duct from the seminal vesicle together combine and form the short ejaculatory duct. The ejaculatory duct opens into the urethra, which is the passageway for both urinary excretion and delivery of sperms in semen into the female reproductive tract. So this urethra is a common path for both urine and the semen. The urethra runs through the penis and opens to the outside at the tip of the penis, which contains erectile tissues derived from modified veins and capillaries. So this urethra runs through the penis, and this penis and this urethra is surrounded by the erectile tissues. These erectile tissues are responsible for the erection of the penis during sexual intercourse. So now here you have a clear diagram of this male reproductive system, and as I said before, here you have the testis. This is the testis. This consists of the seminiferous tubules and the interstitial cells or the Leydig cells. And once the sperms are produced within the testis, it accumulates in this epididymis. This is the long 6 meter long tubular structure. Here the sperms become mature and become motile. So yes, the sperms are stored for a certain while until ejaculation takes place. So once during the process of ejaculation, the sperms from this epididymis move along the spermatic cord or the sperm duct, and here you can see it moves upwards and takes a turn here and here, and then it reaches the vas deferens. This section is the vas deferens, and then it moves downwards, and here you can see the seminal vesicle. This seminal vesicle also releases a part of the semen, and this secretion along with the sperms coming from this vas deferens are together released to this short ejaculatory duct. This is the short ejaculatory duct, and this ejaculatory duct opens into the urethra. This is the urinary bladder, so urine also passes through this urethra, and this short ejaculatory duct also opens into this urethra. And on the way, the secretion of this bulbar urethra or the Cooper's gland is also done into this urethra, through this duct. And then here this urethra extends through this penis and here, and moves out during sexual intercourse. And now here you can see these erectile tissues. This is the corpus sponginosum, and this is the purple is the corpus cavernosum. Those are the erectile tissues. When blood collects into this, the erection of the penis occurs. Next we have the spermatogenesis process. Spermatogenesis is the process of male gamete formation, which includes formation of spermatocytes from a spermatogonium, meiotic division of the spermatocytes, and transformation of the 4 resulting spermatids from each spermatocyte into spermatozoa. Spermatogenesis occurs within the seminiferous tubules of the testis. We just talked about that. The time taken to produce mature sperm cells from a specific spermatogonia in a seminiferous tubule is about 7 weeks from start to finish. So this process takes about 7 weeks, nearly 2.5 months like time. The formation and development of sperms is continuous and inexhaustible in adult human males. So this spermatogenesis process occurs from puberty until generally until the death of the male. Cell division and maturation during gametogenesis occurs throughout the seminiferous tubules. So throughout the seminiferous tubules, the cell division and maturation process of gametogenesis occurs. Hundreds of millions of sperm are produced each day by spermatogenesis, in contrast to oogenesis. So this shall be discussed later. In spermatogenesis, all 4 cells produced during meiosis of each spermatocyte develop into mature gametes. So regarding this cell division process, we'll be talking soon. Spermatogenesis starts at puberty and occurs throughout life. So that's what they said. Development is continuous and inexhaustible in adult human males. Spermatogenesis produces mature sperms from precursor cells in a continuous sequence. So you'll be studying about this general sequence next. The main steps in spermatogenesis. The stem cells. Stem cells are diploid that give rise to sperms. They are the spermatogonial stem cells. Arise from mitotic division and differentiation of primordial germ cells in the embryonic testis. Now, in the embryonic testis, differentiation of primordial germ cells into spermatogonial stem cells occur. These stem cells are situated near the edge of the seminiferous tubules. In mature testis, their progeny moves inwards as they pass through the different stage of maturation. In mature testis, the stem cells divide mitotically to form the spermatogonia. There are two type of cells, which in turn generate primary spermatocytes. They are diploid by mitosis. Then each spermatocyte gives rise to 4 spermatids. Now here, this is the stage of meiosis, meiosis 1 and 2. So each spermatid will be a haploid type of cell. This is the male gamete, reducing the chromosome number from diploid 2n, which is 46 in humans, to haploid, which is 23. So that's half of 46. These spermatids differentiate into sperms with its characteristic head, midpiece and the tail. So this is the structure of the sperm, and we'll be learning about that as well. The sperms are released into the fluid-filled lumen of the tubule, and they travel along the tubule into the epididymis, where they become mature and motile. So this fluid-filled lumen is the lumen of the seminiferous tubule, and they then travel to the epididymis, where they become mature and motile, and where they are stored until ejaculation takes place. After puberty, Leydig cells present between the seminiferous tubules secrete the hormone testosterone, which promotes spermatogenesis. Now, this testosterone is required to promote spermatogenesis. Different cells that undergo spermatogenesis are surrounded and connected to a special type of supporting cells called Sertoli cells. Now, we studied about Sertoli cells, how their orientation is. They are situated on the walls of the seminiferous tubules, and they extend towards the lumen of the seminiferous tubules. So here they say the cells extend from the wall of the seminiferous tubules to the lumen. These cells secrete inhibin hormone and nourish and provide attachment for cells that are in different stages of spermatogenesis. We talked about that. Now here, section A, you have the transverse section of a testis. So here this is the testis, and these coil structures are the seminiferous tubules. They are highly convoluted structures, and through these, once the sperms are produced in these seminiferous tubules, through this vas efferentia, these are some straight tubules, through this vas efferentia, they move on to the epididymis. This is the epididymis structure here. This is a highly convoluted 6 meters long tubular structure. So, and here you can see once the sperms are stored here, they will be released through this. This is the spermatic duct. Next, we have the transverse section of the seminiferous tubule. This is the transverse section of the seminiferous tubule. We studied about that. So here you have the lumen and the various stages of development of the sperm cells are seen from progressing from the wall towards the lumen. And then next you have the spermatogenesis process. This is the C diagram. This is the spermatogenesis process, and here you have the germinal layer. Then you have the primary spermatocytes, the secondary spermatocytes, and the spermatids. And this is the Sertoli cell. Here, these are Sertoli cells. They are supporting cells. And then next D, you have the mature sperms released into the lumen of the seminiferous tubule. So these are the mature sperms which are released to the seminiferous tubule. So here you have the spermatogenesis process in a diagrammatic representation. First you have the primordial germ cells in the embryo, and then mitotic division occurs to produce the spermatogonial sperm cells. These are diploid, and further mitotic division and growth of this cell occurs to produce the spermatogonium. And here you can see this primary spermatocytes. This is the 2N type of cell, and here then you can see the meiosis 1 taking place. During this meiosis 1, the 2N cell divides into 2 cells. These are the secondary spermatocytes, they are haploid cells. And then you have meiosis 2, where each haploid cell divides into 2 haploid cells. This is the second stage of meiosis to produce the spermatids. So these are the spermatids, and then spermatids undergo structural modifications to produce sperm cells. So these are 4 sperm cells produced by the 4 spermatids, and here you can see the head, midpiece and the tail. So talking about the structure of the sperm, each sperm is made up of 3 main parts: the head, the midpiece of the body, and the tail. So here you can see the head, the midpiece and the tail. Head of the sperm contains the haploid nucleus. So this is the haploid nucleus carrying the paternal genetic material. Paternal in the sense the father's set of genetic material. A special vesicle called the acrosome is present at the anterior end of the head. So this is the acrosome. It contains hydrolytic enzymes such as trypsin and hyaluronidase that helps the sperm to penetrate the outer layers of the ovum. So during the fertilization process, you'll be learning that in detail next in the future videos. So for the penetration of the ovum, these hydrolytic enzymes present within this acrosome are required. So that's why they are present at the tip of the head of the sperm. Midpiece of the sperm contains many mitochondria that provides ATP necessary for the movement of the tail. So you know the sperm moves by the lashing action of this tail. So this is an active process, and this requires ATP. So from this mitochondria present between the midpiece of the sperm, the required ATP will be produced. The tail of the sperm contains a long flagellum with the typical 9+2 arrangement of microtubules. It is produced by the centriole found near the base of the nucleus. The tail enables the sperm to swim along the female reproductive tract towards the ovum. So here at the neck you can see the centriole. From the centriole this tail originates. And here you can see the 9+2 structure of the microtubules. Here you can see 9 pairs of microtubules: 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is the 9 set of pairs of microtubules, and in addition in the center you have another 2 microtubules. So that's why you call this the 9+2 structure of the microtubules. So here you can see the outer and inner dynein motor arms and the neck skin bridges. So that's not really important, but this basic 9+2 arrangement can be seen in this tail. They're talking about the semen. The semen is the fluid that contains a mixture of sperms and the secretions of the 3 sets of accessory glands. Now we talked about this accessory glands: the seminal vesicle, the bulbourethral or the Cooper's gland, and the prostate gland. The semen is discharged from the urethra during ejaculation. Usually a normal ejaculate contains about 2 to 5 milliliters of semen, and the sperm count in the semen may be in the range of 40 to 100 million per milliliter. So generally in an ejaculation around 2 to 5 milliliters of semen is released, and so a large number of millions of sperms would be released. Normally the sperm comprises less than 10% of the final ejaculate. So less than 10% you can find the sperms. The rest of the 90% of the semen is the secretions from the 3 accessory glands. Main fraction of the semen is made up of secretions from seminal vesicles and the prostate gland. The semen contains several components such as mucus, enzymes, prostaglandins, ascorbic acid, citrate and fructose, which promote the survival of the sperm. So these are required for the survival of the sperm. So majority of these components are released by the supporting or the accessory glands. The semen provides a liquid medium for the sperm movement and also helps to neutralize the acidity in the female reproductive tract. So as you know, we talked about this in asexual reproduction. We said in asexual reproduction there is an aquatic medium. So here the semen provides this aquatic medium required for the sperm to swim and to prevent desiccation of the sperm. And in addition, they say to neutralize the acidity of the female reproductive tract. So there is an acidity present in the female reproductive tract. This is an adaptation actually. The acidity in the female reproductive tract is an adaptation to prevent the growth of microbes in the vaginal area. So this acidity needs to be neutralized or else the sperms would die. So here these components are able to neutralize the acidity present in the female reproductive tract. The life expectancy of a sperm is about 48 to 72 hours after ejaculation, after depositing in the female reproductive tract. The accessory glands associated with the male reproductive system. There are 3 sets of accessory glands that produce the secretions necessary for sperm survival and movement. They are the seminal vesicle, the prostate gland and the bulbourethral or the Cooper's glands. Seminal vesicles. They are a pair of 2 small pouches that produce a thick yellowish fluid that is expelled during ejaculation. So this produces the bulk of the semen, seminal vesicles. Seminal fluid is alkaline to protect the sperm in the acidic environment of the vagina. It contains mucus, fructose. The fructose provides the most of the sperm's energy, and coagulating enzyme. This helps semen coagulate after ejaculation. Ascorbic acid and local regulators called prostaglandins. So these are the components of the semen that is produced by the seminal vesicles. This fluid contributes to about 60% of the volume of the semen. So that's why I said this produces the bulk of the semen. The secretion of seminal vesicles produce the bulk of the semen. Each seminal vesicle opens into a short duct that joins the corresponding vas deferens to form the ejaculatory duct. We just studied about the structure before. The prostate gland. This gland is found below the urinary bladder. It secretes a thin milky fluid directly into the urethra through small ducts. This milky secretion contains coagulants, anticoagulating enzymes and citrate, which is a sperm nutrient. This fluid contributes to about 30% of the volume of the semen. So now this prostate gland is also important because the ejaculatory duct and the sperm duct open to each other within this prostate gland. The joining of the sperm ejaculatory duct and the urethra occurs within this prostate gland. Bulbourethral gland or the Cooper's glands. They are a pair of small glands found along the urethra below the prostate. These glands secrete a clear alkaline mucus that is able to neutralize any acidic urine remaining in the urethra and lubricates the lining of the urethra. So as I said before, the urethra is a common part for both urine and semen. So remains of urine may be present in the urethra, and as you know, urethra contains uric acid and this can be acidic. So to neutralize this acidity, this alkaline mucus is released by this bulbourethral or the Cooper's glands, and in addition, due to this mucus, lubrication of the lining of the urethra occurs. So this eases the sperms to swim through the urethra. So here you can see the seminal vesicle. This is the seminal vesicle and this is the prostate gland, and then these 2 small glands are the bulbourethral glands. Now here you can see, as I said before, this short ejaculatory duct and the sperm and urinary urethra open to each other within this prostate gland. So this diagram clearly shows the three sets of accessory glands present in the human male reproductive system. Then moving on to the hormonal control or the hormone regulation of the male reproductive system. In males, in response to the release of GnRH from hypothalamus just before puberty, it results in the release of FSH and LH from the anterior pituitary. So GnRH is released from the hypothalamus. GnRH is the gonadotropins releasing hormone. This results in the release of FSH and LH from the anterior pituitary. FSH is follicle stimulating hormone. LH is luteinizing hormone. Rising levels of FSH and LH hormones at puberty promotes mature functioning of the male reproductive organs. The hormones regulate the development, growth, and pubertal maturation of the reproductive process of the body. So that's the function of the FSH and LH along with GnRH. FSH and LH hormones direct spermatogenesis by acting on different types of cells in the testes. First of all, FSH stimulates Sertoli cells to nourish the developing sperm. Then LH causes Leydig cells to produce testosterone and other androgens to promote spermatogenesis. So this is how the FSH and LH contribute to the pubertal maturation and the reproductive process of the human reproductive system. Two negative feedback mechanisms control sex hormone production in males. The first is testosterone inhibits GnRH, FSH and LH in blood through its effect on the hypothalamus and anterior pituitary. Now, with the increasing level of testosterone, testosterone has an ability of inhibiting GnRH, FSH and LH being produced and secreted by the hypothalamus and the anterior pituitary. And next, additionally, inhibin produced by Sertoli cells act on the anterior pituitary gland to reduce FSH secretion. So when the action of inhibin is to reduce the rate of spermatogenesis if it increases excessively. So inhibin produced by the Sertoli cells, they act on the anterior pituitary to reduce the FSH secretion. These negative feedback circuits maintain testosterone and other androgen levels in the normal range within the blood. So here you can see the diagrammatic representation of this hormonal control. First, just before puberty, the hypothalamus secretes GnRH, the gonadotropin releasing hormone, and this acts on the anterior pituitary. First of all, anterior pituitary produces FSH and both LH, and through FSH, FSH acts on the Sertoli cells. FSH is the follicle stimulating hormone. It acts on the Sertoli cells, and the Sertoli cells promote spermatogenesis. In addition, it produces inhibin. Inhibin provides a negative feedback on the anterior pituitary and prevents the production of FSH and LH. And then through LH acts on the Leydig cells, and these Leydig cells are responsible in producing testosterone. Testosterone also promotes spermatogenesis, and this also provides a negative feedback on the anterior pituitary and the hypothalamus to prevent the production of GnRH and FSH and LH. So here you can see this blue color square is the testis, and this section occurs within the testis, and this pink color region is outside the testis. This is related with the brain. And so this is the basic outline of the hormonal regulation of the male reproductive system. So that's the end of our video. So I hope you guys learned something productive and especially also enjoyed this video. And I invite you to like and share this video so that if you learn something productive from this video, even your friends might be able to gain the benefit that you got from this video, and you'll be able to successfully face your A-level examinations. And also don't forget to subscribe our YouTube channel Gamsapi so that you'll be notified as soon as we upload a new video. So thank you very much for watching this video, and good luck and all the best for your A-level examinations.